Keith gave a very nice introduction to most of this. I'm going to talk about that. So my background really is in proteins, these synaptic signaling proteins, and they're called NMDA receptors. Okay, I'll touch on this shortly. And it was because of pure grin that got me very interested in this, that I wanted to start understanding more how to take this to a translational based program. And to do that, I decided to form a collaboration. I actually brought a pointer with me. Hopefully, and I formed this collaboration with a colleague of mine, Howard Surratt, who works on these fish. They're called zebrafish, and I'm going to sort of talk to you today about what they can teach us about NMDA receptors and GRIN disorders, okay? And again, so he's the expert in genetics, I'm the expert in NMDA receptors, and we really formed this very extremely strong collaboration. So again, the general issue is why study zebrafish, right? Here's a zebrafish. So here's a zebrafish. It's actually an adult zebrafish, and we typically don't study adult zebrafish. Again, we're interested in the human brain. We're interested in the human nervous system. We're interested in trying to understand why your children have whatever manifestation they have, right? But we want to use a different system, zebrafish, to address that. And so I just want to bring to you one fundamental concept about biology. And Keith touched on it, and he talked about the alphabet and these things. And I just want to sort of reiterate a few terms, because these terms are important. I'm going to use them periodically. And one of them is the genotype. Okay? You'll hear about your genotype. So in fact, when you got the genetic report, there was a mutation in there, and it was indicating the genotype. Okay, but what was the change in the genotype? And so all systems, and Keith talked about it, there's this DNA, it forms the genes, and that DNA, via the process of going through RNA, will ultimately form proteins. Okay? And the proteins are the part of the system that carry out the function. They manifest what we are, right? And that we're going to call, or people call the phenotype. So you'll oftentimes hear people say genotype and phenotype. And so the phenotype, right, is, my genes, the genes I have in my body dictated my phenotype, which in my phenotype includes my hair color, includes my size of my eye color. My family's got big noses, so that's sort of, I've gotten that from the genotype from my family. Okay? Now, for most of you, the phenotype you care about is what we would call a clinical phenotype, right? Your child has epilepsy, has dystonia, has intellectual disability. So those are phenotype. Those manifestations that you see are manifestations of the genes or the genotype of that individual. And again, so we have, and again, I apologize, I think everybody here has GRIN mutations. I'm going to focus on GRIN mutations, and there are other ones that I'll touch on. But again, the genotype then are these GRIN genes. Okay? And these GRIN genes encode what we're going to be called NMDA receptor subunits. And on the next slide, I'll sort of show that. And these NMDA receptor subunits come together to form a protein, and that protein is called the NMDA receptor. Okay? And that's exactly what Keith was showing. On a synapse, you have a synapse, and you have this little blob and things going through the middle. That's an NMDA receptor. And that NMDA receptor underlies sort of the phenotype in our brain, right? The way our brain controls how we think, how we carry out functions. And I see Dr. Hugener, who's the second speaker, just arrived, and he's going to talk a lot about how those receptors function at synapses to carry out how we work. And one of the challenges is this class of receptors, which is called ionotropic glutamate receptors, is found everywhere in the brain and actually non-brain tissue. Okay? So you name a behavior, you name any bodily function, somehow these receptors are involved in some aspect of it. And that's why when your children have a altered genotype, that one of those nucleotides is changed from a guanine to an adenine, that leads to a pretty severe clinical phenotype because these proteins are so. So here again is just a list of them. This again is the genes. So when we talk about GRIN1, when you talk about your child having a GRIN1 variant or mutation, we're talking about that gene. So just so you know, a little technical detail, the human genes are capitalized. The rodent genes are not, okay? And then the zebrafish genes. So when you talk about GRIN1, that's again the genotype. That's the gene that's encoding a protein, and that protein is called GLUN1. Okay? That's the protein that is generated, and that's the protein that's going to carry out and make the phenotype, whatever that is related to the nervous system function. Again, there's NMDA receptors, there's many genes, there's 7 of them, and including GRIN2A, 2B, 2C, 2D. So you'll hear GRIN2A, and again, that encodes a protein called GLUN2A, and that GLUN2A, again, is going to be involved in the signal functioning of the endocrine. As Keith also pointed out, there's AMPA, these are called AMPA receptors. They have that same GRI, they just have a different name. There's 4 genes here, and then there's kinase. Okay? So these are the proteins that are generated. And I put this in, and I apologize right now. It's a last slide I put in, and I just put it in there because I just wanted to emphasize what these proteins that are generated. Remember the GLUN1 and the GLUN2, that they actually form a protein. And the good news is Keith actually showed you this picture, right? These are these proteins at synapses. Right? And Keith sort of had an image where you could see that things flow across them. So these are these proteins at synapses, and we oftentimes think of these, as I mentioned already, they're essential to all synaptic function in the brain, in the nervous system, not only in the brain, but outside. And here is a researcher, basic researchers. This is the actual image of one of these proteins. Okay, they're using what's called cryo-EM or X-ray crystallography. It doesn't matter. And the only reason I wanted to show you this is here's the protein, and there's 4 of these subunits, and one of them always contains GLUN1. So every NMDA receptor in the body has a GLUN1 subunit. And then they're typically mixed with GLUN2 subunits. Okay? So if your child has a GLUN1, that means every NMDA receptor in their body has, again, they're heterozygous, so you're going to have a wild type copy of non, but you're going to have that. Whereas you'll have wild type GLUN2A, wild type GLUN2B. Okay, so I just wanted, this is information I just kind of wanted to put in there, just to sort of give you a framework. When I talk about the NMDA receptor protein, how it relates, and I talk about GLUN1, how it relates to GRIN1 and GRIN2A. Okay? So now I'm back to my schedule of what I was going to talk about. So I'm trying to introduce some things here just to sort of, again, Keith really introduced some nice things there that sort of laid a foundation. So as a basic researcher and as a clinician, there are some really fundamental questions we want to ask when we discover one of your children have a variant. Okay? And one of the first one is, does the variant contribute to the clinical phenotype? And basically that's the question, is it a loss or a gain of function, right? Because in terms of how you potentially treat that, it's very different whether it's a loss or a gain of function. Again, what a loss of function does is it turns down that activity, or it turns down that signaling mechanism used by these receptors. Okay, I guess I'm gonna give up on this. I'm just gonna sit right here. We'll bypass gravity. So that's a question that's relevant to parents, and it's also relevant to scientists, because we want to understand how that's happening. Now here's a question that's a little more relevant to just a real basic scientist, right? So, okay, you have a variant in the GRIN1, and it causes a loss of function. Why? There's scientists, my lab, a big part of my lab does this. We want to understand why that change in the gene leads to the change in the protein, and how that change in the protein leads to altering the way it functions. Really, it's a mechanical question. Why? How does this lead to it, right? That's not really something you guys are very interested in. Comes down, and the third one you may not be that interested in, but it's really, I think, a critical question. And how does the altered function of the NMDA receptor contribute to the clinical phenotype? Right? How many basic researchers are interested in this question? How does, now we know there's a loss of function or a gain of function. How does that ultimately cause the clinical manifestation that you see? Okay? Why does it lead to epilepsy? Why does it lead to dystonia? Why does it lead to, again, asking really fundamental mechanistic questions, right? How do you lead? And finally, one question I'm going to touch on a little more is, how to treat clinically the individual with the variant. And really the question is to improve the quality of life. And I think attending these Cure-Grin meetings and listening to these, so they always start off, these Cure-Grin meetings always start off with a presentation by a parent. And you always hear sort of, this general theme of the trajectory, what happened. Please can reiterate that with his Braxton. But a big part of it is the quality of life. Finding some way just to make life a little better, a little more normal at that level, right? So we want to have cures, but in many ways, there's a more short-term issue. Many of your families are. Okay, so now back to zebrafish. Why did I decide to go into zebrafish? And I'm going to show you the slide a few times. I'm not going to go through this in very much detail, but I just want to describe today some of the advantages I feel to address using zebrafish to really address these two questions here. Okay? Those are the two questions we want to address using zebrafish. And the first one, and again, there's many advantages, I'll talk about it, but the last one I want to sort of emphasize right now, okay? And that's what's going to be called high throughput drug screens, okay? So one of the challenges with rodent models, again, they're very powerful, these rodent models. They're introducing the variant into a mouse, and now they can look at the phenotype in these mice. But one of the challenges is to do drug screens on this, right? Because again, you could take a mouse and really test only one at a time. Really test one mouse at a time. Whereas zebrafish, because they're incredibly small, and because they have really robust behaviors, we can do it on a much larger scale. Okay, so this here is I was looking at this and I realized it's not something many of you would be familiar with, but every scientist knows what that is. It's a 96 well dish, right? It's just little individual chambers, and actually I'm going to show you on the next page what it looks like. So that's a dish. We can put 96 zebrafish in there, and we can assay 96 drugs at a time. Okay, not one, 96. And that's illustrated here. So again, they're using actually a stimulus response. It's a behavior. So they're assaying a behavior, and that behavior is a representation of some part of the circuit. Right? It could be epilepsy. It could be pre-pulse inhibition, which is an index of schizophrenic behavior. I'm going to show you another behavior shortly that can relate to this. But what they can do, and you can see, I think it's still, I don't know if it's still, you can see there's flipping through there every now and then. Anyway, I don't have access to it. But you can do it on a much larger scale, right? And so, again, this is one of the advantages of Zebrafish. I think it froze the computer. Movie. Oh. Yeah. It's contacting. Oh, there. I think maybe I solved it. Maybe I didn't. Yeah, it is frozen. Yeah, frozen. I'll just wait. There, I got it. Okay. So again, this strategy has been successfully applied to various treatments, including Dravet syndrome, which shows indices of epilepsy. Okay, so this overall strategy has been used in Zebrafish, and sort of our, one of our overarching goals is again to take Zebrafish, insert the variant into Zebrafish. We're using a new technique, it's brand new technology, and we're now successful in it, to do a behavioral assay, and then to be able to do high throughput drug screens, either on clinically approved drugs or small off-molecule libraries, to see if we can alter in some way that phenotype, that clinical phenotype, and then ultimately to take anything screened through here initially to mice, and then hopefully ultimately to be able to recommend it to a physician. Okay, again, this is sort of one of our overarching themes here as we continue to move forward. And that's illustrated here. So again, we want to take the fish, and we're introducing these variants into them. So again, we're changing the select little bit of alphabet that Keith talked about, and then to see how that affects them. And again, we want to ultimately do drug screens. In addition, or in parallel with that, we also want to carry out with Zebrafish these sort of more mechanistic studies. How is it that this variant, this change in the gene, is altering the circuitry? How is that altering the circuitry, and how is it affecting the way the nervous system functions? And again, here, our idea, and we're certainly not the first ones to come up with this idea, is the idea of identifying novel therapies. And I'm going to show you one of the ideas that we have. And again, these are just some of the variants we now have going. This animal is now being completed. We have it fully being tested. These are a lot of GRIN2B variants we're now working on, just to get them going. Okay, so I don't remember how my schedule was. It was
Lonnie, could you go back one slide?
Yes, I could do whatever you would like.
You wanted to take pictures.
Oh, you guys want to take? Again, these are they're just variants that we're now working on, and we're not really tied in with a patient or anything. We've selected them for various reasons. Right? But, again, this, as I told you from the beginning, or when I talked a little earlier, you would like the trajectory to be at this rate, and science at this rate, right? So again, there's some things here. So again, advantages of Zebrafish. So what are some of the advantages? Again, external fertilization and rapid development. I showed you a picture of an adult zebrafish. We rarely work with adult zebrafish. Almost everything is done with larvae. Okay, so it speeds up the whole process. So again, here's the timeline for the development of the zebrafish. At time 0, there's an egg, and that's done externally. So we can do manipulations on that if we like. But within 3 days, you reach the end of what's called the embryonic development phase, and then you move into what's called post-embryonic development. So you get an animal that looks like this. It's still, early on, it still has the yolk sac, but these animals can carry out really complex behaviors. So during this whole time in here, they're carrying out complex behaviors, and therefore we can assay various features of these animals. And within 2 weeks after they're born, we can start carrying, or 7 days after they're born, we can carry out assays to start studying both drug screens as well as basic mechanisms of how this is changing. Okay? And I'm just going to show you one well-characterized larval behavior. So to start this whole project, to start this whole collaboration, we took an approach, and what we did is we deleted the genes, the endogenous genes in zebrafish, one by one. So we knocked out the GRIN1 gene. We knocked out the GRIN2. And then we studied those as a sort of a template. So when we start having a GRIN2A variant, that's sort of our framework to start studying. So here's a behavior. It's called the social interaction behavior. And it's really a very important behavior because it's a hallmark for neurodevelopmental disorders. So in fact, autism is typically diagnosed based on behavior. That's one of the major criteria to diagnose. So what is our assay here? So this is the first bit of science you're going to get here. So here's a lane, okay? And here's a lane, and there's a zebrafish there. And then at one end of the lane, we have a window, and we have another fish there. That's a conspecific, so it's the same animal. And then at the other end, we don't have anything. And we put the fish in there, and it can decide where it wants to go. Okay, it can go down here, it can go up here. And it's known in wild type zebrafish that they don't care who they hang around with at young ages, and that's illustrated here. So they're just kind of randomly selecting things here. By 14 days or 2 weeks post-fertilization, they're still kind of randomly, although you start seeing they're spending more time up in zone 4, and by 3 and 4 weeks post-fertilization, they're now want to be around animals like them. Okay? So that's called a social interaction. They are actually picking to be associated with their conspecific. So they're spending, as they mature, they spend more and more time close to their conspecific. So it's just an assay. Again, it's a behavioral assay. We can do this very rapidly. We can turn it over. Right now, we only have a chamber for 10, but there's a program that we're trying to get a hold of where we can do up to 48 assays at one time. So this is to do a larger program. And here's what's very interesting. If you look at the GRIN2B fish, again, which is a high-risk gene for autism spectrum disorder, so you see that they lose completely this whole phenotype. So GRIN2B fish, and it was actually quite surprising, GRIN2B fish are pretty normal. We were kind of sad about it. They were pretty normal. Turns out they have a lot of compensation. A lot of GRIN1's upregulated, GRIN2A's upregulated. But whatever upregulation they had could not substitute whatever the key role this gene was involved in this phenotype, this social interaction phenotype. And so, again, this is just another representation of that same data. I'm not going to go through. Okay? So I just want to illustrate that's one aspect of the advantage of zebrafish. We can now assay a behavior that's very relevant to the clinic and to the brain circuit. So now the next real big advantage of zebrafish is to say, okay, where at and why is that happening? Why is this sort of behavior now somehow changed? And again, I'm not going to, so this is zebrafish. Their brain really does not look at all like humans, but there are many regions that share homology, such as the pallium and the subpallium, or telencephalon, and they're part of the forebrain, and there's analogous structures in humans. But what you can do is, again, taking advantage of the genetics of zebrafish. There are fish that they express red only in those cells, those neurons that Keith mentioned, that use glutamate as a neurotransmitter. And then there's another marker, it's green, that expresses this green protein, you don't need to know the protein, in those neurons that express GABA. Okay? So now we can go ask the question, what part of the brain is changed in these Grin2B fish? What part of the brain is presumably underlying this altered phenotype? And when we did this, we discovered, I give a different version of this when I talk to different audiences, I forget that. And we discovered it's in this subpallium, okay? This region right down here, that we see a loss of a particular population of neurons. And these are called VLX. Again, it's a transcription factor, but it's related to GABA. So if you recall, Keith mentioned that, the terms of trying to identify drugs, a big part is to identify those that target the NMDA receptor, but another part is to target those that don't target but target associated systems. Okay? And what we've identified here, at least initially, there's many more things we're trying to do, is that this region's in part of the social interaction circuit, it's been shown before, but it's also the birthplace of a whole class of interneurons. And we speculate now that in these fish, that this protein, Grin2B, is important to the development of this completely different class of proteins that we hadn't thought about being important to the phenotype. Okay? And so again, as Keith said, maybe that we have to think about not thinking about targeting glutamate receptors, but maybe we have to target a different system that might facilitate those children with that phenotype. Okay? Again, this is just sort of the general plan. And I want to show you one last thing here. And this is an area we're really excited about. The problem is Howard and I are neither experts in it. And it's called, and this is another advantage of zebrafish. And you're going to see how relevant this is to the family shortly. And when we think about NMDA receptors, when we think about this whole class of ionotropic glutamate receptors, we think about them at synapses, right? We think about brain function. And when we went into this, that's what we were really interested in, is how does these change in the genotype lead to changes in brain function. In the process of doing this, we had a very talented PhD student. She's now a postdoc in my lab. She made a very important observation, and that the lack of NMDA receptors was not only affecting the brain, but it was also affecting cell types related to what's called the neural crest. Okay? And the neural crest is a bunch of different populations of cells. Again, I don't want to get into the details, but it's just a lineage, right? As the brain develops, there's different lineages that form, and some of them form the lungs, some of them form the heart, and there's one lineage called the neural crest, and we saw an alteration in those. And the neural crest forms things like facial bones, melanocytes, so these give color, Schwann cells, these are part of the nervous system, but most of these are not in the central nervous system, but outside there. They involve the teeth, adipose tissue, but the one I think that we're very interested in is the enteric nervous system, because when we hear the parents talk, one of the big questions is gastrointestinal issues, right? Constipation, eating problems, right? So it's possible, again, we have no direct evidence for this, we're trying to move forward with this, that the development of the neural crest is part of the mechanism that's disrupting the enteric nerve, and I should say the enteric nervous system, again, I apologize, I'm using a terminology, is what innervates the gut. So that's the nervous system that innervates the gut, okay? And again, these are just some of the Molly has identified. You know, we see changes in facial structures in those animals that don't, the fish that don't have the NMDA receptor. We see changes in the melanocytes too. So again, these are both types of tissues derived from the neuroplasm. So we're very interested in this question. How is the change, the role of NMDA receptors in neuropress leading to altered features of sort of the non-central nervous system, not the brain, but rather the sympathetic nervous system, which is also very important in these neural storms. But mainly we're focusing right now on the interior system or the gut, to develop assays, behavioral assays, to be able to monitor gut motility and see. So this is my second to last slide. Again, just your take-home message. I just want to emphasize we've done a lot of analysis. We think zebrafish are a really positive model to study human disorders. We're not the first ones to come up with this idea by far. I mean, this has been a powerful system. One of the strengths is these high throughput drug screens. It offers the opportunity to identify compounds at a more rapid rate. You can identify mechanisms underlying the development of phenotypes. We think these other type of neurons might be important or might be related to GRIN2B disorders, these GABAergic neurons, and to try to develop novel approaches to target them, to target the critical phenotype. And finally, zebrafish have really sort of offered a venue to start studying sort of something that we normally don't think about, cell type that we normally don't think about, being related to the cell. Okay? And again, I want to acknowledge Howard Sorokin. He's the geneticist in the group. Again, I study, as I talked about, these proteins, NMDA receptors. And these are the people. Molly is now a postdoc. She was the one who discovered the NeuroCrest. Josiah Zuzman was the first graduate student. He was a joint graduate student between us. He's now at UPenn doing a postdoc. And Erica Nevitz, a new MD PhD student, and she's actively involved. The rest of these are involved. Okay, that's it. Any questions? I think. Any questions? Sorry if I use technical terminology sometimes. I try to avoid this.
I have questions, but I don't know if I'll remember the technical terms.
You can use whatever you want.
The picture of the thing that, in my mind, looks like a Connect 4, with the you said it was 96, and you could put 96. Yeah. I don't know if that thing was called.
Yeah, 96 well played. I don't know if I want to go back to it. But
that's fine, you don't have to, but I guess my question was, did you say that could be scaled? So,
well, typically,
or that you can only do 96 at a time.
Typically 96, but you could do, you could imagine you have multiple. That's kind of a unit there, right? Yeah. Again, it's going to depend on the behavior that you can assay, right? So we can't, for the social interaction assay, we can't use that. We can't use a 96 well. That's okay. So, but there's a number of behaviors in zebrafish that you can detect that with the high. You have to have a high-speed camera above it to track their movement. Okay.
So when you talk about doing the drug screens, what is the timeline for that? Like, is that just an ongoing thing that because there's a gazillion drugs to screen?
Well, I mean, obviously we've been screening some drugs, but we really haven't pushed it up. In part because the variants themselves are now just coming online. So again, you have, it's not like we can take this. It's a very new technology that we're using to introduce individual new changes in this alphabet that to turn it to the variant. So we're creating what are called these missense mutations, right? So just a single nucleotide over the millions of them I don't even know if there are billions of nucleotides in the chromosome. And we're able to convert one of them to match the variant that is in each other. And we're continuing to try, and we've got it going. So working. We've got now our first one on Lonnie, but we're continuing to try to figure out ways to optimize it. The system is you know, there's templates, Cas9, different. And once those start becoming, you know, a more common feature, then we're really going to start pushing more of the drugs.
So I heard you talk about how you can make changes for like, I can't remember what it is, but like the variant. You can do a variant, but can you do a deletion?
You could, so actually, and I know your son has an exon 6 deletion, and I tried to look at it, and deletions are actually more straightforward to do, right? Because you can use this, there's a standard technique you can use to just introduce a deletion. The problem with doing that in zebrafish is not the technology is complicated, and we certainly could do it. It's more how it's processed, right? And so I don't fully understand how a cell handles deletion in one of two alleles, right? Remember, every gene has got two alleles. That's the two copies of that protein. So there's actually two genes for green tumor, and they're called alleles, right? They're called alleles. And the missense or the deletion is only occurring in one of those. You have a wild type copy, and so there could be really complex interactions between them. There's things called nonsense mediated decay. It could be, so there's a lot of things I don't necessarily know how it would handle. And a geneticist would really have to be involved to do the experiment. But technically, it's a more straightforward experiment than everything.
Thank you.
Yes, Scott.
So will the zebrafish change the normal paradigm starting with, a short cell going to a mammalian cell going to a mouse? Where along that will it work?
Well, I think in terms of old general mechanisms, it's pretty clear evolution has been fairly, in terms of basic mechanisms, evolution has been pretty conservative. So the basic mechanisms are probably going to be common across all species. But in terms of the drug screens, I mean, the advantage of zebrafish is we could do a high volume, but then we're going to get some positive hits. But really, to get it closer to the human, we first have to probably do it in a mouse, if that was your question, I think. And then how it looks in a mouse, where it's a lot more limited, then you would go suggest it to the doctor and decide.
So you're getting, not getting rid of the mouse, but you're getting rid of the frog or the...
Well, no, the mouse, again, the problem with mice in terms of drug screens is one, it's incredibly expensive, right? Incredibly expensive, and it's low volume. You can do one mouse at a, I mean, nowadays the behavioral chambers have greatly expanded, but still, you can do 10 mice at once versus 96 fish at once. And the fish, you can test them at 5 days after birth, right? And they absorb things across their gills, so you don't need to inject them, you don't need to handle them. There's just many ways in terms of the volume. And it gives you an initial framework to think about what you might be able to use, and then you should take that with the mouse where now you're testing 5 mice. That's the general idea.
What type of medication is used to treat this?
It's going to vary depending on, it's going to depend on whether it's loss or gain of function. It's going to depend on, one thing I didn't talk about, loss or gain of function, it's a big protein, right? So a loss or gain of function could be very different, whether it's in this end of the protein or this, lead to different clinical features. Where it's at. Okay?
Yeah, so when you talked about, we knock out the genes completely.
Yeah.
So that would give you something kind of approximating or some kind of loss of function.
Yeah.
So then if you create an individual, and then like a knock-in where it replicates the patient variant, you'd still need to do the phenotyping on whatever the variant is. You could do the phenotyping on the gain of function to see how it's different from other fish. and then identify something that's different, and then try to do the different drugs?
Yeah, well, so, I mean, so what we're trying to do, so we're actually doing two strategies with coming back to this. One, we're introducing the variants into the zebrafish gene. So we're introducing the modification. So we're taking two strategies. One strategy is that zebrafish have their own genes, right? And we're introducing, we're looking at that part of the alphabet that they have. It's basically the same as humans, and we're changing that in the zebrafish genes. And that's one thing we're looking at, okay? And then, you know, initially here, we want to see the phenotype in these animals, right? Do they show a phenotype, right? And can we get, you want to be able to see a behavioral phenotype, really. And so what do we do? A bunch of assays, prey capture, social interactions, lots of different assays that we now have developed in the last 3-4 years that we can assay these. Okay? And once we see a behavioral alteration, then that gives us a template. Because right, we gotta be, to do drug screens, we have to be using a behavior. Right? And then that gives us a template at which we could do the drugs. Now again, in parallel, we'd certainly be interested in how does that affect the way the brain develops. Okay? That's one strategy. The other strategy that we've been working on is taking the background of the fish without a gene. So we take a fish without GRIN2A, so the GRIN2A genes have been knocked out in the zebrafish, and we're reintroducing into them the human GRIN2A. Okay? And that's progressing, but again, science moves slowly. And then the goal would be to able to humanize the zebrafish using the human genes, and then also to use the human variants, right? So that, okay. Again, everything, you know, these are a lot of experiments that we're talking about here, and making progress. And any other questions? So I'm going to turn it over, I guess. Is there a time break?
Yeah, we'll have a break now.